So what are we talking about today? Today we're going to go ahead and focus on gene expression and how do we regulate that, right? So the first question that we really want to ask ourselves is, why do we regulate gene expression? We're also going to look at both prokaryotes and eukaryotes because the methods that they're using are slightly different. So within prokaryotes, we're going to go ahead and look at how bacteria, which is a prokaryote, use an Operon system, and we'll specifically look at the trip operon and the lac operons and how those are regulating expression of specific genes. We'll then go ahead and look at how eukaryotes are going to regulate gene expression and focus mostly on how that process is being regulated through transcription, and then what are some things that are happening after transcription to also regulate gene expression. And finally, we're going to talk a little bit about a pretty new technique that's called RNAI, which is standing for RNA interference, and how does that involve microRNAs, and how is that also regulating gene expression, because that's also happening after transcription. So let's go ahead and start in with why do we regulate gene expression. So here on the left, you can see a fruit fly, right? And you can also see here that within development, right, in these different areas, different genes are expressed kind of, right, at different times within development. So that's one reason why we see that genes are expressed differentially, right? Because expressing certain proteins is involved in development, right? You can also see over here kind of within human tissues, right? I mean, essentially within every cell that's in your body, right? It has essentially the same genome, right? So the same set of genes. And the reality is, though, it is about most cells are using only about 20% of those genes kind of at any one time. Right, I mean, as cells get even more specialized, such as in nerve cells, muscle cells, and things like that, they're using even a smaller percentage of those genes, right? And so regulating gene expression is going to allow us to go ahead and differentiate the tissue types, right? So that's also going to be important. So let's kind of start with thinking about bacteria and how this is working. So one thing that's kind of important to think about is like, how can you use gene expression to regulate the production of a metabolic compound, right? So this is an example of the synthesis of tryptophan, right? And so trip, when we're talking about trip operons, right, that's really standing for tryptophan. Okay, and what we're seeing here is there's actually two ways to kind of regulate this metabolic pathway. One, right, we have something called feedback inhibition. Which we've talked about previously in the year, right? And so if you remember, right, in that case, here's the product, right, which is tryptophan. And when there's a lot of tryptophan, right, that's going to go ahead and is going to bind to the enzyme. And as it does that, right, it's going to alter its ability to kind of like catalyze that reaction, right? So it's most likely going to go ahead and bind to an allosteric site, right? It's going to go ahead and cause that enzyme to change shape. Since its shape is now different, its structure, since its shape and structure are now different, its function is different, and it's no longer able to effectively catalyze tryptophan, right? And so since the pathway stops here, right, you're no longer creating more tryptophan. So this is a fast way, right, that cells could go ahead and turn down the amount of tryptophan that they're making, right? And this is something that's beneficial, right? Because you're not then making excess, committing like resources to make excess tryptophan than you really need, right? But another way, which we're going to look at in more detail, is really through gene expression, right? So as you regulate gene expression. So you could also, right, instead of going ahead and blocking one of these proteins, right, by altering its structure, you could also go ahead and essentially turn off all of these genes, right, because there are 5 different genes here that are going to be involved in making these different enzymes within this tryptophan pathway. So that's going to be a second way, right, which is going to take a bit longer, right? This is a little bit slower response, right? But this is another way to go ahead and regulate the production of tryptophan. And that's what we're going to kind of focus on a bit today, right? So how is this working within bacteria? Well, bacteria are using what are called operons, right? And so operons have a couple of different regions, right? So if you have the promoter, right? If you remember, the promoter is where the RNA polymerase is going to bind, right? So this is where the RNA polymerase binds to go ahead and start transcription, right, to create the mRNA that you need. Okay, and then if you notice, right, there's a couple of other genes that are going to be important in here as well. You have the operator, and the operator is basically acting like a switch, right? And it's going to go ahead and essentially say, all right, is this on or off, right? And then you have, right, so really these, and then you have the genes that you're going to transcribe, right? So these are the genes that you're going to transcribe the mRNA of, right? And one thing that's really important to kind of like think about is you can put all of these genes and group them together, right, within an operon, okay? And so that's what we're seeing with like tryptophan, and we'll see with lactose as well. By positioning them like this, right, it's going to go ahead and allow you to make all of those products at once, right? So you would make all of the enzymes in that tryptophan pathway all at the same time. And that's advantageous, right? So there's another, so together, right, this promoter, operator, and these genes that you're going to transcribe, these are effectively called the operon. Okay. You also have this regulatory gene, and we'll talk about that in a little bit more detail. But this regulatory gene, this is going to go ahead and it's going to create the mRNA for a protein that then is going to regulate the operon, and it's going to do that by binding at the operator, right? So what we'll see in some examples as we think about this, their proteins will bind at the operator, right? Will bind at that switch and can essentially go ahead, and that's how we're going to regulate if it's on or off based upon if a protein is present or not, and it's really going to depend kind of on the model. But this is what we're seeing within bacteria and within prokaryotes in terms of how they're regulating gene expression. So let's go ahead and take a look at two different examples. We're going to first look at the trip operon, and then we're going to go ahead and look at the lac operon. So let's go ahead and take a closer look at the trip operon. Right, so this is a good look at the trip operon, and you can see here, right, you have the promoter, right? So this is again where that RNA polymerase is going to bind. You have the operator, which is acting like a switch, right? You have these different tryptophan genes that are coding for these different enzymes, right? So you're going to create the mRNA for those different enzymes so you can create those proteins all in the same pathway, right? Because those enzymes are going to allow us to create tryptophan. And then you can see upstream of this, right, you have this repressor protein, right? And so this repressor protein is always being made in the cell, but it's really being made in a small amount, right? But what happens, right, is that it's also being made inactive, right? So that it's really unlikely that it's going to bind and really like have a high affinity for the operator. So what's going to happen here, right, is you see that so when there's a lot of tryptophan, right, the tryptophan is going to go ahead and bind to the repressor protein, and it's going to activate it, right? And so now you have this activated repressor protein, right? And so its shape has changed, right? And so now its shape has changed, and since its shape has changed, it can now go ahead and bind to the operator. And since it's going to now bind to the operator, right, in this case, now RNA polymerase can't go ahead and go past the operator, so it's not going to be able to transcribe those different trip genes, right? So at this point then, none of those enzymes are being made. You're effectively turning off this pathway, and then no tryptophan would be synthesized, right? So if we go back and kind of look at this in just a little bit more detail, right? And think about this again, right? So now this has something bound to it, right? It's now going to be able to go ahead and bind at the operator. This can't pass it. This mRNA is not made. None of these proteins are made, right? And no tryptophan would be made, okay? And so one thing to think about that's really important is this essentially, right, until the repressor binds, transcription is really happening all the time. Right, unless the repressor is bound. Okay, and so there's two different types of operon, and since it's working in this way, we call this a repressible operon. right? It's repressible since this repressor is going to go ahead and bind to the operator and cause gene expression to be turned off. So this is a classic example of a repressible operon. Okay, so now I'll go ahead and take a look at the lack operon after you kind of see an animation of this, right? Because an animation will kind of help tie this together. Then we'll go ahead and take a look at the lack operon.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is inactive. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription. The tryptophan repressor is a helix-turn-helix regulatory protein. When tryptophan is absent from the environment, the repressor is in an inactive conformation and cannot bind to the DNA to prevent transcription. When tryptophan is abundant, 2 molecules of tryptophan bind to the repressor. This alters the orientation of the helix-turn-helix motifs in the repressor and causes their recognition helices to fit into adjacent major grooves of the DNA. Thus, the synthesis of tryptophan occurs when it is needed, but is repressed when tryptophan is available.
Okay, so let's go ahead and take a look at the lack operon. With the lack operon, this is an example, right? So a trip operon is what we call a repressible operon. This is what we call an inducible operon. And this will make more sense in a second. So this is an inducible operon because here's what's happening. So what we're seeing is that normally, right, transcription is off. Right? This is the normal kind of default, right? And so when we're looking at lack operon, we're really talking about the sugar lactose, right? And lactose in this case is really being broken down into 2 sugars, right? Because what you're going to be able to do is split lactose to make glucose and galactose. So, but if there's no lactose in the cell, then there's really no need to make these different genes, or to make the different proteins, right, from these different genes that are going to allow us to break down lactose. So let's kind of look in terms of how this is going to really work, right? But the first thing to understand is it's really starting in an off mode. So what we're seeing is this. So when there is lactose in the cell, you want to be able to turn this on. So it is inducible, right? It can be induced, it can be turned on. So when there's lactose in the cell, there's a lactose, right, which is an isomer of lactose. Okay, so when there's lactose in the cell, there will also be a lactose because that's an isomer of it, and it will go ahead and bind to the repressor, right? And when it binds to the repressor, right, it's going to cause it to go ahead and come off, right, of the operator, right? So the repressor was bound before here, and now it's going to go ahead and come off, right? And since it comes off, now RNA polymerase can go ahead and move down the DNA, and it's able to transcribe these 3 different genes, right, that are part of the lack operon. And again, the benefit of the operon is allowing you to transcribe all 3 of these genes at the same time, making all 3 of these proteins, right? And all 3 of these proteins are going to be involved really in the breakdown of lactose, okay? And so this is, again, an example of an inducible operon, right? And it's inducible because you're able to, let's spell that again. So this is inducible, right? Because what you're able to go ahead and do is induce transcription, if you will, right, by adding lactose. Okay, but something else to understand that's important is both the lack operon and the trip operon are both an example of negative regulation of gene expression, right? And these are negative regulation because the repressor, right, when it's in its active form and is bound to the operator, it's turning these off, right? So these are both examples of negative regulation because the repressor, right, when it's active, binds to the operator and turns these off. So now let's go ahead and take a look at an animation of the lac operon and how it works to kind of further clarify this.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the lac operon. The unit consisting of the lac promoter, lac operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the lac operon. Located near, but not in tandem with, the LAC region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the lac repressor. In the absence of lactose inside the cell, the lac repressor protein is active. In its active state, the lac repressor recognizes and binds to the lac operator site. When the lac repressor is bound to the lac operator site, RNA polymerase is prohibited from recognizing the lac promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the lac repressor. This causes a conformational change in the lac repressor protein that prevents it from binding to the lac operator site. Inactive lac repressor is unable to bind to the lac operator site. Without an active lac repressor bound to the lac operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the lac operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
So we looked at 2 previous examples with the TRIP and lac operon involving negative gene regulation, where the active repressor is binding to the operator and causing gene expression to be turned off. And now we're going to go ahead and look at positive gene regulation, which is also happening at the lac operon at the same time. So within the cell, right, so we're really talking in bacteria here, right? And so within this bacterial cell, what we're going ahead and seeing, right, is that it wants to go ahead and use glucose, right, as its energy source, right? Glucose is its preferred energy source. That can then, right, go into glycolysis, right, and that's going to allow it to synthesize ATP to go ahead and run the cell, right, all of the different functions that are within it. So if there's not a lot of glucose present, though, right, this E. coli, right, this bacterial cell, will also go ahead and it will use lactose, right, if it needs to, right? But again, it prefers to use glucose. Like, glucose is much more preferred, right? So what happens, though, right, when glucose levels are low, what we end up seeing, right, is if glucose levels are low and if there's lactose present, this molecule that says CAMP, right, they're made in high levels. So you're going to have a high level of this. And CAMP really is standing for cyclic AMP. Okay. Okay, and what it's going to do, right, when you have high levels of cyclic AMP, this is going to bind this CAP protein, which is standing for catabolite activator protein. Okay, so this is going to go ahead, right, and bind to the DNA, right? So this is binding at the promoter here. Okay, and what it actually does, right, is this stimulates the binding of RNA polymerase. Okay, so let me kind of recap that, right? So when glucose levels are low and there's lactose present, you want to go ahead and turn up the lac genes essentially, right? You want to go ahead and be able to take and break down lactose. To do so, what's happening is there's higher levels of cyclic AMP in the cell. Those will go ahead and bind to this inactive catabolite activator protein, right? And as that happens, it becomes active, right? Its shape changes, okay? It will go ahead and bind to the promoter, and that's going to facilitate the binding of RNA polymerase to the DNA, right? And that's going to allow it to go ahead and transcribe all of these lac genes, right, which are going to allow us to go ahead and take and break down lactose. Okay. So the other thing is too that still has to be the case, right? You still have to have the repressor, right, has to be removed from the operator, otherwise this doesn't happen, right? So generally, right, the RNA polymerase, it will, still bind, right, if the repressor is off, right? But it's going to bind. It actually has a much lower affinity, so it'll bind and make these lac genes at a much lower rate versus if you go ahead and have high amounts of cyclic AMP. So this is a way for you to basically kind of like turn the volume up, right, or turn the knob and go ahead and say, man, I need a lot of these proteins that are going to go ahead and allow me to break down lactose, and I need to make them fast, so I'm going to go ahead and turn this up. Okay, and so you can kind of see this, right, when you go ahead and remove, right, so that there's if there's a high amount of glucose, right, you're going to go ahead and you don't really need to use lactose, right? And if that's the case, now there's no cyclic AMP, right? And if there's no cyclic AMP binding here, right, at that catabolite activator protein, what we're seeing is, right, this is inactive. It will not go ahead and bind to the promoter, right? So RNA polymerase will still bind, but at a lower rate, right? And so you're going to go ahead and go and make these genes at a much lower rate. So this is an example of positive gene regulation, right? Because going ahead and having this catabolite activator protein bind to the promoter, it's really stimulating the expression, right, of these lactose genes that are going to allow us to eventually break down lactose. So before, when we were talking about the TRP and LAC operon, we were talking about how we're regulating gene expression in prokaryotes. So now let's go ahead and take a look at how that's a little bit different when we're talking about eukaryotes. So if you can see here, let's kind of blow this up and look at this. What's going to happen is some sort of signal is going to come in, right? We're going to kind of focus on what's happening in a couple of spots, right? So we can go ahead and we can regulate expression through transcription. We can also regulate it through how we process these transcripts after, right, they're produced, and then we can also think about, right, the processing in terms of, right, so here we've processed it, we've spliced it, okay. Then we can also look at capping, because capping is also another way to regulate gene expression. Basically, the length of this tail affects like how long this mRNA sticks around in the cytoplasm. And then we can also go ahead and see as well, right, that once it's in the cytoplasm, we can regulate it after that point as well, right? We can go ahead and see that mRNAs are degraded, right, and actually broken down, and we'll look at that when we look at RNAI. And you can also see that proteins get chopped up sometimes with proteasomes, and then, right, so there's a lot of just different ways that we can regulate gene expression within eukaryotes. And this is going to allow us to have like differential gene expression, right, within different cells, since, again, muscle and nerve cells are super differentiated, right, and on average we're using like 20% of the genes in the cell or less, right, depending upon how differentiated it is, and again, its function is going to dictate kind of which genes are turned on, if you will. Okay, so if we're looking at the structure of a gene, right, in terms of a eukaryotic gene, again, you have this promoter, right, and this is where this RNA polymerase is going to bind. Okay, right? So you have these RNA polymerases that will bind, right? Because that's going to allow us to go ahead and transcribe our mRNA, right? And that mRNA will then allow us to build our protein. Now, if you notice here too, you have these control elements, right, that are sitting near the promoter. Okay. Now, basically, and we'll look at this in some detail, these control elements are where you're going to have transcription factors bind, and that's going to become important. Right? So you're going to have these different transcription factors, and they can either be activators or repressors, right? And obviously this is going to allow you to go ahead and you can have all these different ones binding. There are often many different sites, and since there are many different sites, it's going to allow you to go ahead and either turn up or turn down, right, the amount of mRNA, specific mRNA that you're going to go ahead and make. Okay, so when we're looking at transcription factors, they're like, if they're an activator, they're going to go ahead and bind and promote the binding of RNA polymerase, right? So that it's better able to bind. It's only going to work when all these transcription factors have bound. Then it will bind, and then it will go ahead and kind of move down and synthesize the mRNA. Right, and so that's what we're seeing here, right? Is that the transcription factor is binding at this binding site, which would be upstream of the promoter. Okay, and as it's doing that, right, these transcription factors are regulating which genes are expressed, right, and so which proteins are made. Okay, and that's really important, right? That these transcription factors really are going to help with the regulation of the binding of the RNA polymerase, right? So the RNA polymerase, its binding is affected by these different transcription factors. Okay, and that's something to go ahead and focus on.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So once the transcript has been made, right, so you had your transcription factors bind, if they were going to go ahead and activate it, right, we're seeing that transcription has occurred. And now there's some things that we can do after transcription to go ahead and also regulate gene expression. So the first thing is thinking about splicing, right? So remember, in splicing, you're having introns that are being removed, right, and exons are being joined together, okay? And one thing, right, is you can have this alternative splicing where, you know, different exons can be spliced together, right? So you can get different combinations, and this is again what allows us to essentially, right, produce multiple proteins from a single gene, right? Because if we have only 23,000 genes, we have way more than 23,000 proteins. So that's one way we're regulating this, is by splicing these exons in different combinations. The second thing is looking at this untranslated regions, right? So you have these UTR regions, okay, sitting right before the caps, right? And so for these UTR regions, right, because those are after the start and stop codons, so they're not translated. But these are also really involved in regulation of transcription, right? Because typically, if this is larger, right, if you have a larger UTR region, the mRNA takes longer to degrade, right, to break down. And so if it's going to take longer to break down, it's going to be in the cytoplasm longer, right, which means that you would see more of that mRNA binding to ribosomes, right, more times, so then more of that protein would be made. So that's one way to go ahead and regulate that. The other thing is oftentimes, too, proteins can go ahead and bind, right, onto these UTR regions and regulate translation, right? So make it so essentially so that, because these are important for binding to the ribosome, and so since you're going ahead and having these proteins bind, it's going to prevent binding of the mRNA to the ribosome, and since the mRNA is prevented from binding to the ribosome, you're also preventing translation from happening. So this is two other ways, right? So by looking at alternative splicing, also looking at, right, the fact that you have these UTR regions, right, that if they're larger, it's taking the mRNA longer to break down, it's present longer in the cytoplasm, so you're going to go ahead and see more of that protein being made, right? And then also you can essentially turn this off by having regulatory proteins bind in this UTR region and making it so that this transcript won't bind, right, to the ribosome and then not be translated into protein. So a couple of other ways that we're also regulating this after transcription, right? And we're going to look at one last thing. We're going to look at a technique called RNAI. So it's used quite a bit now in modern biology, but this is also another way that gene expression is regulated after transcription. So the last thing that we're going to look at here is a technique called RNAI, which stands for RNA interference, right? And so what you're seeing here is these miRNAs, right? And miRNA is standing for microRNA. And these are single-stranded. So what you're seeing here is your primary microRNA, and these have these hairpin loop structures. And then what actually happens is there's this enzyme called dicer that essentially cuts these, right? So it cuts these into chunks that are about 20 to 22 nucleotides long, right? And so it chops it up, and what happens, right, is there is the actual microRNA piece, right, which is only one of the two strands, okay, because again, it's going to be a single-stranded piece. And so you have this microRNA that binds to a protein. And what happens, right, is when this microRNA binds to the protein, this can go ahead, right, and bind to a specific mRNA, right, because this has a sequence. The microRNA, right, has a sequence, and if it's complementary, right, to this mRNA, it will bind to it. And when it binds to it, it's going to go ahead, right, and with this microRNA protein complex, it's going to go ahead and basically break down this mRNA. And if it's going to break down the mRNA, right, then you're not going to have translation happen, right, so then that protein won't be made. And this could be used, right, in molecular biology because you can essentially target a specific gene, right? You can go ahead and say, well, okay, what's the mRNA that it's going to make? Well, we can go ahead and we can use the microRNA transcript that would be complementary to that, and then, right, it's going to go ahead and if we put it in the cell, it will go ahead and break down all of the mRNA that would be complementary to that, right? And so that will allow you to target a specific gene, and you can essentially knock it out, right? In that way, right? And go ahead and knock down the expression of it. So this is another way that we're seeing that's a little bit more unique. This is a little more recent, right? Since about 2003, we're seeing this. Another way that we're regulating gene expression in eukaryotes after transcription. So as you can see from this lecture, right, there's a pretty big difference in terms of like how we're regulating gene expression in prokaryotes versus eukaryotes. And so we'll finish this off with going ahead and looking at an animation in terms of how RNAI works. And I hope that this is kind of showing you how gene expression is being regulated both in prokaryotes in terms of using operons, and then in eukaryotes thinking about what's happening before transcription with transcription factors, thinking about what's happening during transcription, thinking about what's happening really after transcription, because there's a lot of different ways that we're regulating gene expression, right? Because we want to turn up and be able to turn down different genes at different times for a lot of different reasons.
RNA interference is a mechanism of silencing gene expression after the gene has been transcribed. This interference is due to the presence of antisense RNA, which is complementary to a specific cellular mRNA. The complementarity between the antisense RNA and the cellular mRNA causes the two molecules to bind to each other. The antisense RNA can come from three sources. First, it can be introduced into cells by researchers. Second, certain viruses may produce antisense RNA as part of their life cycle. And third, the integration of genes into chromosomes next to endogenous gene promoters can sometimes cause the synthesis of the antisense strand of RNA. The current model of RNA interference involves two steps: creation of an RNA-induced silencing complex, RISC, and then the degradation of cellular mRNA by the RISC. Creation of the RISC occurs when double-stranded RNA is found in the cell. This double-stranded RNA is recognized by an endonuclease known as dicer that cuts it into pieces that are 21 to 23 nucleotides long. An antisense RNA fragment can then associate with proteins to form the RISC. The silencing complex can then associate with cellular messenger RNA that has a region of sequence homology with the RNA fragment in the RISC. The messenger RNA is then degraded. The presence of this rapid degradation mechanism can reduce and even effectively eliminate translation of the messenger RNA for the gene in question, thus interfering with gene expression.